Now we are going to talk about photosynthesis. Photosynthesis is the formation of chemical energy stored in the glucose bonds and oxygen releasing. Also, it is in charge of anabolic reactions and formation of molecules. This is all in presence of sunlight. The sunlight that plants, bacteria, and algae use to make photosynthesis is a set of waves called spectrum, and they have different wavelengths. Thanks to this difference, they are classified into three categories: the UV rays, that are the rays we can see, the vision range, that are the light we can see, and the infrared rays, they are the radiation of heat. In plants, there's a thing called photosynthetic pigments, and they are like things that capture the solar radiation through our photopigments, and they are also responsible of coloration. And talking about this coloration, there is a thing in plants called chloroplast, and it absorbs different wavelengths like the violet, blue, and red, and it reflects green. Carotenoids absorb blue and green and reflects yellow, orange, or red. Phyocyanin absorbs green and yellow and reflects blue or purple. We talked too much about concepts. Now we're going to talk about action, process, stages. The first stage is the light dependent reactions. It happens when sunlight is transformed into chemical energy thanks to two people, ATP and NADPH. It has two systems. The first one, it helps to collect the light, and the second one to carry the electrons. Carry the electrons. Talking a little bit about the second stage, it is called the light independent reactions. It is when those two carriers that help in the first stage, ATP and NADPH, are dissolved in the stroma. It has this C3 cycle that it recollects CO2 using our UVP, and this CO2 will be released by the plant, and it rises 12 molecules of 3C atoms of PGH to make glucose. And the C4, it also captures CO2, but instead of 3, it has 4. But there are some things that can affect all the process we talked before, like sufficient water or CO2, low T inhibits speed, or finishing the amount of nutrients, the age of the plant, the leaf shape, and the chlorophyll content.
We can analyze, and also we can perceive that two things. One that is almost imperceptible in the video is that the flame began to make a noise like a whistle and became a little bit brighter. The plant that we had was a family of the algae, but it was not specifically the algae, so it took like more time, and in this case it took 2 hours approximately to see how the plant made the photosynthesis, and with the light that it receives, it can receive the inorganic matter and transform it into the organic matter in the algae. We can see this with little bubbles, but because of our plant require more time when we did the experiment in the laboratory, we only saw one little bubble or two maximum.
During this experiment and stage, we learned about photosynthesis and how some plants and algae can perform this to transform light into energy for themselves and then in oxygen for the rest of living beings. In my opinion, it's a complete experiment about all the things, and it's irrefutable about the generation of oxygen within the perform of photosynthesis in a plant, specifically in an algae.
Hello, I'm Karla and I will talk about the cellular respiration and its importance for the living beings. Cellular respiration is a fundamental biological process that occurs within the cells of living organisms. It is an increased series of metabolic reactions that converts glucose and oxygen into energy, carbon dioxide, and water. This energy, in the form of adenosine triphosphate (ATP), is the primary fuel that powers various cellular activities essential for life. Through the stages of glycolysis, the Krebs cycle, and the electronic transport chain, cells extract high-energy electrons from glucose molecules and shuttle them through a series of proteins, ultimately generating ATP. This process is crucial for the survival and function of all living beings, as ATP is the universal energy currency that powers activities such as muscle contractions, nerve impulses, and the synthesis of essential molecules. Without cellular respiration, organisms will lack the energy needed to carry out the vital processes, leading to a breakdown in their biological functions and ultimately their survival. Thus, cellular respiration stands as a cornerstone of life, demonstrating its profound importance in sustaining the diverse array of organisms that inhabit our planet.
Our first step for this experiment is to pour some yeast and sugar inside the test tube with warm water. And then we take the sugar
and we mix. 3 layers may still be observed, but the substance that used to be transparent is now translucid, and there are solidified parts of yeast and sugar at the top. After 10 minutes of mixing, the mixture finally dissolved.
with this balloon. We need to stretch it and place it at the top of the test tube.
It has been over 15 minutes and it finally has started to inflate.
It still can't get up on itself, stand up on itself. But there's a little bit of gas inside the balloon already. It has been 20 minutes since the experiment started, and the balloon finally rising up. It has already been 30 minutes, and this is how our balloon looks like. It is inclined from this part, and it's still growing. It has finally been an hour, and this is how the balloon looks like right now.
To analyze the behavior of the release of gases in the experiment, both a qualitative observation and time are required. Something that can be said at first sight is the increase of the size seems to be exponential. We divided the experiment in 4 stages. The first one characterizes by a 15 minutes time lapse, in which gases were slowly being released from the mixture into the balloon. The second phase characterizes by a 5 minutes time lapse, in which the balloon finally could stand up on itself from the pressure inside itself from the gases released. Then, the third stage was pretty much as the last one. It continued an exponential growth in its size for a period of 10 minutes. Finally, an hour had passed, and it was time for the fourth stage. In this part, since there was not an infinite provision of glucose for the reaction to repeat infinitely, the reaction rate decreased. This does not mean that the balloon decreased in size, but that it didn't increase as much as in the previous stages.
In the previous experiment, we saw an explanation of cellular respiration through a simple home experiment that involved materials that can be found everywhere around. As we know, cellular respiration involves the breakdown of sugar with the purpose of turning it into ATP, which is called adenosine triphosphate, that helps energy to make its basic functions. In this case, the extension of the balloon represents how the mix of yeast, sugar, and warm water produces a carbon dioxide gas as a byproduct. Our experiment turned out good, as we can physically see how the gas is stored in the balloon and has some bubbles formed in a glass tube. The experiment successfully represents the basic functions of glucose in organisms and its importance.